No. We'll be starting in another 30 seconds from now, Dr. Cole.
Good afternoon, ma'am.
Good afternoon. Good afternoon. I think we're all set to start.
Right.
Hello. Testing, testing. 1, 2, 3, 4, 5, 6, 7. Somebody here is logged in. Login again. We'll start on count of 3. 3, 2, 1. Start. On behalf of the Department of Neuropathology, it's a pleasure to welcome you all to this webinar on the art and science of special stains in neuropathology. It's a special welcome to the President of ICMLS, Mr. Kaptan Singh Sehrawat, and Dr. Pankaj Kaul, who's Secretary of the ICMLS, which is the Indian Confederation of Medical Lab Technologists. I think if a pathologist is asked to name the single most crucial factor which is necessary for the success of a lab, I think we'll all unanimously vote for the technical staff. And our department, I think, is blessed to have an excellent group who are not just skilled, but also very committed, and to have instilled those virtues of quality, dedication, and commitment. Much of the credit goes to our chief technician, Mrs. Mary Kashi. She has been with us since the, I think, inception of the department itself, and has contributed no less than 38 years, 5 months, and 26 days of dedicated service to this department. Her contributions to the growth and development of this lab are enormous. I must say she's an expert in several fields, but her mastery in special stains is most notable. And I think the reputation that we enjoy, the compliments our department faculty receive for all our beautiful pictures and presentations and publications, we actually owe to her expertise. But I think it's her passion for the art that has captured all our hearts. And even more importantly, she's an excellent teacher. Several technicians have been very lucky to have been trained by her, so that many more can receive the benefit of her experience. And to honor her enormous contribution to the service of this department, we thought it is most befitting to have a webinar on special stains. And to make this available to the technologist community in our country, we approached the president of ICMLS, Mr. Kaptan Singh Sehrawat, who readily agreed to disseminate this information amongst their members. And I think it's a pretty huge response that we are looking at when we see the YouTube. So Mary, this is dedicated to you, dedicated to your service, and most importantly, the teaching. In this platform, Mrs. Mary will be sharing her expertise with all of you with a very detailed video of each step, right from reagent preparation to the actual procedure. But I think the highlight will be the panel discussion, where she will be sharing the useful tips and tricks of the trade that she has learned over decades of her experience here. We sincerely hope all of you will benefit from this webinar. So I think, I'm sure you're all very excited to start. We will begin with a very basic understanding of the principles of stains used in neuropathology and their application. I invite Dr. Nandish, Additional Professor of Neuropathology, to begin this webinar.
Thank you, madam. Thank you very much. And very good afternoon to all of you. Namaskara. Yeah, in the next 10-15 minutes, I will give you an overview of the special stain utility, its principles and application. To start with a few introductory remarks, I'm going to the grassroots level and then coming to the actual application. So histology, we all know that it's the scientific study of tissues. Of course, tissues are made of cells, so you can extend it to cells. Now, if you add patho into that, that is a scientific study of the diseases affecting the tissues or the cells would be histopathology. Now, we all know that any particular disease, we need to have scientific evidence. So for that, histopathology plays a very crucial role. It forms a pillar, foundation, cornerstone for the diagnostic evaluation of many, many diseases. Now, if you want to look at the tissue and cells, it's colorless, transparent. So we need to highlight different components of the tissue or the cells. And to have the contrast, we need to do the staining. Now, if this staining utilizes the principle of chemical reaction, then it will become an histochemistry. And this chemical reaction is either utilized from the tissue property itself, then it will become an histochemistry. So basically, the chemical reaction can happen within the cells or in between the cells, or you can create a cellular reaction by adding some kind of bondage linking to that. Now, what is important here is that when histopathology forms the essential component of the diagnostic workup, then the technical work involved in that histopathological section, staining and the evaluation forms a very crucial integral part of this. So what I would say is that if there are no stains, no histology, no histopathology. Now, stains form a very important technique or technology wherein we can visualize various components, biological components, cellular, tissue level. So why do we use the word special for these stains? Now, anything other than routine, you know that routine staining is hematoxylin. So after the initial evaluation of hematoxylin, whatever staining technique you do to pick up any particular element, particular property, particular component would be called as special stain. But my definition would be a little beyond. Special stain is a staining performed by special person. So, of course, for us, every technologist is very special. But for special stain, since they try to recognize the special character of that particular staining, a special character of that particular tissue or the section, so these people are generally very special. Now, this I already told you. So now special stains have become an indispensable part of histopathology laboratory and immense aid to the histopathologist. Now, we should not forget, we are a poor country. Of course, there are rich people within that poor country, but since still we are resource restrained. So in a resource restraint, I think special stain will be of great aid. We cannot afford big things. So even in research, we can utilize this special stain. Now, like clinical medicine and evaluation will lead to investigations, histomorphology supported by routine and special stains will aid in or will help in choosing the ancillary test or addition test. So what we need to stress here or reinforce or recognize here is there are many phenotypic markers, like based on morphology and morphological characters, we can pick up many markers, which you can call as biomarkers because they define a biological property. And to recognize these biological properties or phenotypic markers, special stains play a very huge role. Now, why special stain? What is its role? Now, if you take up histopathological evaluation, it still requires, in a sense, HND cannot fulfill many things, so you need some additional testing, especially to recognize some specific features, a specific property, or more precise results. So I'll just put in across a few statements which are more like kind of descriptive poetry you can take. Everything means one or the other way interlinked, so they're all same meaning one or the other way. So basically, it helps in evaluation of tissue, assist in decision making, of course, HND also, but that will be the first step. The next step would be the special stain. Next, augment tissue morphological analysis. That means to say it helps in going into the more deeper part of the morphological analysis, formulate differential diagnosis, then confirm the diagnosis what you would have made on HND, then helps in recognizing the severity of the disease or tissue or prognosis. That is, if fibrosis is there, generally indicates very chronic, maybe it's kind of advanced disease like that. Now, as I mentioned to you earlier, to select the next set of tests, immunohistochemistry or anything, this can aid. And there are many things which you can demonstrate on this, many cellular elements which looks alike or similar on HND. And most importantly for organisms, most of the organisms, as you said, as we all know, we call microorganisms. Of course, it will be more micro because they are just with microscope, you will not be able to recognize. So they'll be special. So you need special things for that. Of course, not to forget in the boundary of neuropathology is the enzyme activities for muscle enzyme histochemistry. And most similarly, many abnormal deposits, we cannot differentiate, we cannot pick up on routine staining. So you need to depend on special stain. And in neuropathology, why is, of course, this is our specialty. Even otherwise, the greatest challenge, maybe I'm biased because we are working in the field of neuropathology, but in general, if you take up right from anatomy, all of you in your first year would have studied anatomy, physiology, biochemistry like that. I would always feel that neuroanatomy is difficult, neurophysiology is difficult, natural neuropathology. Even in clinical medicine, neurology would be the most tough. The reason is this controls every other organ. It's a very unique organ. So what a lot of lesions which occurs can manifest like other organ manifestations or other organ problems. And this tissue has a wide variety of changes which are different from other organs. So you need to have a good backup or a good kind of armamentarium of workup or diagnostic studies. So whatever it is, the lesions have a lot of diagnostic pitfalls and challenges. So you need to equip yourself with many special stains and additional workup or ancillary testing. Now, why special stains in neuropathy? A few examples I'll tell you, then I will go into the applications. Now, in this, as I'm mentioning the utility of special stains, I will be addressing some of the examples. After that, I will just give you a very principles of very essential special stains. Then we can go on to the video demonstration by Mary Menon. Now, in general, we know that many tissues, many structures appear similar on H&E stain sections. Like if you take up collagen, it looks same as compared to amyloid. So to pick up amyloid, you have to depend on special stains, that is congruent serious stain like that. Of course, beyond that, you have to go for birefringence, polarization. Next, vacuoles. Many conditions cause vacuoles. It could be accumulation, it could be some kind of degeneration. To recognize what is accumulating, we need to do special stains to pick up whether it's a fat by oil red bow, Sudan 3, Sudan 4, Sudan black like that. And glycogen storage disorders, phosphoric acid shift, polyglucose and bodies like that. Next, if there is any alteration in the tissue, like for example, stromal excess, fibrosis, all those things, we can do Masson's trichrome, which is a connective tissue stain. We can do Van Giesen. Again, Van Giesen for fibrosis. Silver stain, I will come to silver stain. Elastic stain, very important to recognize the blood vessel abnormality, especially the artery, because they have the internal elastic lamina, elastic stains. Now, another important thing is many of the things indicate, as I mentioned, prognosis. So to recognize the stroma or alteration architecture, we may have to do reticulin. For example, in a pituitary adenoma versus pituitary hyperplasia, we depend on reticulin. Masson's ring, of course, to pick up fibrosis. Of course, you can extend it to liver for chronic liver disease, liver fibrosis, to recognize capsule. Myxoid stroma, the stroma, to recognize the property of the stroma, we can do some of these stains like Alcian blue, musica, carmine like that. Very important. Now, many of the diseases in brain affect the myelin. The myelin is very essential, not just for nerve conduction, even for the integrity, both functional and anatomical. For this, we need to depend on special stain because routine H&E stain will not be staining this myelin. Of course, microorganisms, other than parasite, we will not be able to pick up any, except few which are naturally pigmented, like chromoblastomycosis. On that particular fungi will have natural pigment. Other than that, it's very difficult to pick up any of the microorganisms. So you depend on special stains like Ziehl-Neelsen stain for mycobacteria, Zn stain, modified Zn stain for nocardia, lepra like that. PS, Gomeris, methenamine, silver, musicarmine, all these things belong to fungi. Of course, musicarmine, Alcian blue are specifically for cryptococcal fungi. Giemsa, toxoplasma like that. Of course, some of the fungi you can suspect like mucor, even aspergillus, but to confirm, you may have to depend on special stains. Phosphotoxic acid, Nematoxin, fibrin, we'll discuss that. Next, to recognize deposit, like iron, melanin, everything looks similar. So you have to do special stains to pick up which particular pigment is getting deposited. And not to forget, in the arena of neuropathology is muscle, to be totally depend on special stains in the evaluation of muscle biopsy. Now, going further, we all know there is no need to mention this, that is accurate diagnosis is very crucial. Not only accurate diagnosis, timely diagnosis is also important. Now, a lot of advancement has happened, at least in central nervous system. We are totally dependent on immunohistochemistry and molecular advancements have happened, but still special stains is a very crucial part in our day-to-day practice of histopathology. And I would say that this will complement, support immunohistochemistry or guide towards immunohistochemical stain selection. Now, few more things about principles. Now, the principles are very wide. So very few things I will highlight so that when we pick up select group of stains, it will be helpful in picking up which particular principle is working for that. Now, this is very easy to understand. Direct staining or this direct staining is based on the affinity property. So you just put it some particular component has got affinity, like glycogen has got affinity, like that. Some of the things. Next is dissociation. Like a chemical or compound, if you put, it will get kind of dissociated either into two ions or two components, either acidic or basic. That will react with the tissue and give rise to color result, chromogenic change. Next is based on the electrostatic property, where dyes will specifically go and bind to the opposite kind of charge or like that. Another one is covalent binding, a strong bondage between that. Charged colloids, where positive will go and attach to the negative. Solubility, like this is simple, you can recognize already, it dissolves in lipid. Otherwise, lipid will get dissolved in the routine processing reagent, which we use like xylene, other things. So we can use the property of solubility for, especially for lipid stains. Enzymatic action, this is basically, it is utilizing the property of enzymes which are present in the tissue that will convert into an unstained compound into a stained result, which is based on the enzymatic property. So the end result will be a colored product. So that will give rise to coloration, or the end result, end product will change the chromogen into a chromogenic substance result. That is the one. So the entire enzyme histochemistry is dependent on this particular principle. Argentine reaction is another one. Next is basically, the next is chemical reaction. There is active enzyme, substrate, and then end result or byproduct which is coming up. So pyruvic acid shift, that whatever we use for fungi or glycogen is based on this one. Pth Stain also is based on chemical reaction, like the acid, wherever it is present, it will liberate spheric current that which is present in hemosidin. That is the one which reacts with the potassium ferrocyanide, which is there in the Prussian blue reaction or Pth Stain. That will give us to a blue end product, that is spheric ferrocyanide. And lastly is the dye deposition. This and the first are somewhat similar, but not all will actually allow dye to deposit. It should be relatively stable. To allow this dye to deposit in a very strong way, you can use mordants. Mordants is basically which firmly fixes some kind of chemical to that or dye to that. So it's more like a covalent linkage which is formed. So just first, I am starting with the simple technique and ending with the last technique. These are the things. But pertaining to CNS, there are a few things you should remember. One is the acid-base reaction and oxygen reduction reaction. This we'll understand a little because most of the stains which are used in CNS pathology is based on these two principles or chemical reactions. That is, silver stain, if you take up, it's based on the principle of where silver ions or its salts or compounds will get deposited on the target tissue. It needs two things. One is some kind of neutralization, some kind of sensitization, and next is reduction. Only in the reduced state only it can go and deposit. Now these silver particles are of varying sizes. They accumulate and they deposit in different areas of the tissue, and that will be analyzed based on their parameters. That is, based on the size, configuration, and the number and the size of the silver particles, it will give different tinge or different hue of the black color or silver color. So based on that, not silver color, black color, based on that you recognize. So it's very important recognizing the axons, nerve fibers, and deposits, especially like tangles in degenerative disease, senile plaques. These are very important silver stains. Of course, now you have immunohistochemistry, which has taken over some of these things because these are quite laborious and really it tests the skill of the technologists. Now, next is important one is which can be used. Of course, it goes beyond light microscopy. Of course, light microscopy, it is in between light microscopy, that is semi-thin sections where we use the property of metachromasia, but this can be extended to other evaluation also. That is, tissue will alter the conformational structure of the dye and you get a different color other than the original color of the dye. So the best example will be toluene blue and cresol violet, which will help in recognizing this kind of metachromatic property of these substances. Now, there are a big list of, actually, already I've just listed a big list of special stains which are utilized in neuropathology. Of course, main stressing is on this. Of course, not to forget is the myelin stain, which we'll be discussing, taking examples. So since the list is very big and time constraint, the very crucial ones which are of great use have been selected. So we cannot go into the in detailed aspects of some of these stains. We have selected only 4 or 5 stains, and on that only we'll be discussing the principles, application, and even the video demonstration will be on that. But after the panel discussion, you can ask any questions beyond these stains also. So the best first example of a special stain would be Nasal Stain, which is of great utility in neuropathology. These are basically basic dyes. That means they stain the basophilic part of cells. Say you call it as basophilic because they affinity to basic dyes. That's why, and they can the nuclear ribosomes, they're all basophilic in nature. And neurons have Nusselt substances, which are nothing but collections of rough endoplasmic reticulum or RNA. And the density of these Nusselt substances variable in different types of neurons. And the RNA content, based on that, you get different density or the strength of the basophilia. Okay, so these are actually stained. They have got their RNA is very basophilic, so they have affinity to the aniline dye, and basically Nusselt stain itself is an aniline dye. So you get this color. With variation pH, you get a degree of differentiation. So different cell have the capacity of differentiating in a different way, reacting in different pH. So the result is that Nusselt substance which are present within the neuron will have a dark blue to purple, which will help in recognizing the Nusselt granules. Now, what is the application? Of course, they are present in the neurons. So it's in neuropathology, it's a great utility in demonstrating the neuron and neuronal pathology. So it will stain it will appear as clumps of stain deposit, clumps of ribosomes, which represent the clumps of ribosomes. So they are basically rough endoplasmic reticulum. So any change in the density, distribution, all those things will indicate the pathology affecting the nerve. So it will indicate neuronal loss, neuronal damage, neural degeneration, like that. And of course, secondarily, indirectly, you can also assess the damage of the tissue, which can be extrapolated to the axons and myelin, like that. The next, again, is a myelin stain. It's a Luxol phos blue stain. Here, it's actually an alcohol counterpart of Alcian blue. In the sense, Alcian blue is very water soluble. This is very much similar to that. That's why blue is ending. So it's an alcohol soluble counterpart of this. So this contains actually alcohol soluble amino salt of sulfonated copper, phthalocyanine. Very complex name, but we just need this first line, I would say. The mechanism is simple, acid-base reaction, as I mentioned to you earlier. So there is salt formation because of this acid-base reaction. And brain is very much rich in lipids. Many are lipoproteins. They are basic in nature. So they replace the base of the dye and you get a color reaction, which is usually blue or bluish green stain. The result is that depending on the basic nature of it, the base nature of it, you get different uses. Myelinated fibers are the one which we'll be testing for. It will give a blue color, like as you can see in this application. One second, so this is what is in the white matter. So I'll go back to that. This is the myelin. So with myelin loss, you will see that there is loss of this blue color, indicating myelin loss. So I'll just go back to just to show the photograph I was telling. So myelinated fibers will give a blue color. That's a white matter, which is rich in myelinated nerve fibers. You will get blue color. So other cells will give different colors of blue. Nerve cells will give a purple color. If you have used crescel violet, with yours in the background will be light rose, as you can see in this particular diagram. Okay. So now the utility of Luxol phos blue is mainly for myelin. One good thing is it's quite resistant to light, heat, acids, and alkalis. So you can preserve the way you preserve HND slides. So myelin stain, it's very important that many of the pathologies of brain are dependent on myelin, either primarily affecting the myelin or secondarily affecting. So this is a very great use stain, which is of great use in neuropathology. And one more good thing is this doesn't need a mordant. That is, you don't need a bondage between the dye and the tissue. So this directly acts, so you can apply on, just like HND you can do on formalin fixed tissues. The next stain is actually, again, a myelin stain, but you do on peripheral nerve. The principle is reduction of chrome salt to chromium dioxide by the myelin. So this myelin reduces the chrome salt to a compound that is chromium dioxide. And this chromium dioxide will act as a mordant for the hematoxylin, and it will entrap it and will give a very intense color, a blackish, brownish black kind of thing. So the myelin ring is staining. As you can see, it will highlight the myelin pathology. The background will be light rose depending on the stain and counter stain. Then is the application. Application is mainly, since it's a myelin stain, of course, it will help in demonstrating or picking up the myelin pathology. So there are very wide variety of myelin pathologies affecting the nerve. So whether it's a primary myelin pathology or secondary, all those things you will be able to appreciate. Then not only that, indirectly we can also recognize the nerve fiber density, but we can only pick up the myelinated nerve fiber density, their distribution, their pattern of loss, all those things. So next is phosphotungstic acid hematoxylin. This actually, as the name suggests, a mixture of phosphotungstic acid and hematoxylin. Now this phosphotungstic acid, as the name suggests, has a lot of tungsten elements. So this can bind to hematin. Our tissue has got a lot of hematin, which are iron derived, and brain is one of the important, brain and muscle, they are rich in iron. So once they react, they will get a blue lake pigment. This blue lake pigment actually stains the various structures in that muscle cross striations. One, fibrin is one, another one is nuclei. So now rest of the phosphotoxic acid stains red brown to the rest of the elements like connective tissue, collagen like that, and gives a different color. So you have a contrast. So that means single stain is giving two color. So you can use polychrome. Many of the stains are polychrome. You don't have to specifically say for this. Now, what is the result? You can do on all these structures, and all these structures give different color, but what we are interested is mainly in the striated muscle fibers, where we'll use it for recognizing the cross striations as in this case. So any change in the myofibrillar network, the architecture of the individual muscle fiber or the myofibrils within the muscle fiber can be appreciated. Any alteration in the structure of these myofibrils muscle fiber can be recognized. Lastly is the lowest method of staining. We know that Kulczycki path needs a secondary fixation. So only after that you can perform that. If by chance, if you have missed that and if you have processed it, then you cannot do Kulczycki path. So on a formalin fixed paraffin embedded tissue, you have to use lowest method of staining. The principle is like this. Here the tissue has got, since you have not done a secondary fixation, you may have to mordant with iron alum. Then this iron salts, what it does is it reacts with hematoxylin and that forms a hemat. Now this hematin has got affinity to myelin. So indirectly, you instead of post fixation, you are taking the step like this and you are staining the myelin using the same hematin which is formed using the iron salts in this particular Loeys method. So what is the application? So the same application as KPAL, that is Kulczycki path, which helps in picking up the myelin pathology, myelin rings. Only thing is you do on formalin fixed paraffin embedded. Since you are doing a formalin fixed paraffin embedded, some limitation will be there. It will not be as crisp as Kulczycki path stain. So many things we have discussed. So just to summarize, at least we should remember that special stains helps in remembering, highlighting three things. One is chemical character, the composition. It could be an ion, it could be a metal, or it could be a protein, abnormal protein that like biological character. There is various pathologies it can pick up microorganisms like this. Next is, that is it. Now we are going very high. A lot of advancements are happening in the field, but we should not forget our background. So the entire background or the stability with which we stand is because of the basement and the pillar and the foundation. And that foundation is histopathology. And the foundation for histopathology is the technology. The technologists work like that. So histopathological assessment is a crucial part in any pathological evaluation of tissues. Of course, going beyond diagnosis, going into prognosis, prediction, and research. And stains, as we mentioned, integral part of histopathology. Without that, we cannot work. And despite advancement, some staining methods we still depend. There is no replacement for it, I would say. Now, of course, we cannot depend on single stain. We have multiple stains, and this forms one of the important element. And what is important here is whatever expertise we have or whatever best microscope we have, the staining is not good. Nothing goes further. And this depends on the efficient technologist, their skill. So whatever theory will not work here. It's purely ability of the technologist, commitment of the technologist, and the special nature of the technologist. As Anita Madam mentioned, the pillar of any laboratory would be the technologist. And I would say salutations to entire team of technologists who are involved in many, many fields. And that's how the subspecialty have come. Whatever subspecialties have come, everything are below this umbrella. And we respect them, salute them. And most importantly, we kind of bow down and enormously thank and express our gratitude our technical pillars, that is our technologists. Thank you so much. If you have any questions, we can discuss at the end during panel discussion. Now we'll go ahead with the video demonstration of those 5 stains which I mentioned the principles. Okay, over to the video demonstration.
Neuropathology is very colorful with many stains to demonstrate different structures in the nervous system. In this video, I'll be showing you 5 of the most useful and commonly used stains in neuropathology. For each stain, I'll be showing you the reagents used, how to prepare the reagents and do the procedures. At the end of each stain, I have given a few practical tips and tricks from my experience. Kulczycki pall or the K-pall stain is an excellent stain to demonstrate myelin in a nerve. Reagents required for Kulczycki pall K-pall stain. First is Fleming's fixative, which acts as a moderant, and second is K-pall solution, and third is 0.25% of KMNO4, and fourth is 2% of oxalic acid. Now we'll see how we prepare the Fleming's fixative. 1% of osmium tetroxide 1 ml and 2% of chromium trioxide 9 ml and glacial acetic acid 2 drops. This is prepared every time freshly and it has to be used on the same day. Now the nerve segment is cut into 3 bits. 1 TS, 1 LS for routine processing, and the best 1 transfer section in Fleming's, which has to be kept at overnight, and after 24 hours it has to be washed well in running water and taken for routine processing for K-pall stain. Now we'll see how we prepare Kulczycki pall solution. Dissolve 1 gram of hematoxylin powder in 15 ml of absolute alcohol. Mix thoroughly. Add 1 ml of glacial acetic acid and 85 ml of distilled water. Solution is stable for 3 to 4 months. Now we'll prepare 0.25% of KMNO4, which is used for differentiating the myelin. Take 0.25 grams of potassium permanganate, dissolve in 100 ml of distilled water, store in amber color bottle at room temperature. Now we prepare 2% of oxalic acid, which is used for bleaching KMNO4, 2 grams of oxalic acid, dissolved in 100 ml of distilled water, store in any clean bottle at room temperature. Now let's look at the procedure for the K-pall stain. Now deparaffinize your sections with 2 xylene and different grades of alcohol and bring it to water level. Wash in tap water. Incubate the slides in K-pall solution for 2 hours at 56 degree in an oven, or microwave at level 1 for 30 seconds, which is a preheating, and you can incubate at 56 degree in an oven. After an hour, rinse in tap water. Rinse with distilled water. Treat with KMNO4, which will differentiate the myelins for 30 seconds to 1 minute. Check under the microscope if the myelin rings are seen clearly. Otherwise, you can go back for the same step with KMNO4 for another 15 seconds to 30 seconds. Check under the microscope each step. Then treat with 2% of oxalic acid, which will bleach the KMNO4. Check under the microscope if the myelin rings are seen clearly without any background. Rinse with distilled water. Check under the microscope. Counterstain with usine for 1 minute. Dehydrate with 2 changes of alcohol. Then clear it with 2 changes of xylene and mount with DPX. The mounted slide, when it is looked at in the microscope, the myelin rings will stand out black and the background with light rose of usine color. If the contrast is black and pink, that means your stain is perfect. Few tips and tricks for K-pall stain. The commonest problem that you face with K-pall staining is that it doesn't work sometimes. The most common reason is that the osmium you use in Fleming solution gets oxidized. To prevent this, osmium comes as 1 gram vial, so make stock solution of 10% by dissolving 1 gram of osmium in 10 ml of distilled water. This can be stored as multiple vials, each containing 1 ml in the freezer. For the working solution, take the first vial and dilute with 9 ml of distilled water to make 1% of osmium tetroxide. The second trick is the time of incubation in K-pall solution can be reduced from 2 hours to 1 hour. To do this, preheat the K-pall solution in microwave at level 1 for 2 minutes, or if you don't have a microwave, you can incubate in an oven at 56 to 60 degree for half an hour. Llb stain is useful for demonstrating myelin in a nerve biopsy when you receive only a paraffin block, for which we require 10% alcoholic hematoxylin. That means prepare 50 ml of alcohol and 5 grams of hematoxylin powder dissolved and stored in brown color bottle, which is stable for 6 months. Second is 4% iron alum. Dissolve 4 grams of ferric ammonium sulfate in 100 ml, which is stable for 1 year, stored in brown color bottle. Third is saturated lithium carbonate. 1 spatula of lithium carbonate dissolved in 100 ml of distilled water, which is stable for 1 year, which can be stored in any clean bottle. Now we will prepare Llb's hematoxylin working solution, which contains 10% of alcoholic hematoxylin 3 ml, distilled water 45 ml, saturated lithium carbonate 2 ml for faster ripening. This has to be prepared fresh every time. Now let's see the steps for Llb stain. Deparaffinize your slides with xylene, alcohol and hydrate to distilled water. 4% iron alum is used as a mordant, minimum of 4 hours. Rinse with distilled water. Place in working Llb's hematoxylin solution overnight. After checking the stains the next day morning, check under the microscope. Then if the myelin rings are seen clearly, then you can counterstain with usine if required. Or if you feel that the myelin rings are not seen clearly, differentiate with 4% iron alum. Check under the microscope. Repeat this procedure until the background is clear. Then counterstain with usine. Dehydrate, clear and mount with DPX. When you check under the microscope, the myelin fibers or the rings stand out as black and the background light rose with usine color. The tips and tricks for Llb staining is, although we have said that Llb solution should be freshly prepared every time, if you store at 4 degree, it can be reused for maximum of 15 days. And the second one is, if necessary, check under the microscope and then differentiate with 4% iron alum. Luxal fast blue is used to demonstrate myelin sheath in brain tissue. We require, first is Luxal fast blue solution, second is 0.5% of lithium carbonate, third usine, fourth 0.25% of chrysalect violet. Now we'll see the reagent preparation. Luxal fast blue 1 gram dissolved in 95% of alcohol 100 ml and 10% of acetic acid 5 ml. Mix and filter the solution, which is stable for 1 year. Store it in an amber color bottle. Second is 0.5% of lithium carbonate. Lithium carbonate 0.5 grams dissolved in 100 ml distilled water. The third solution is 1% usine and the fourth is 0.25% of chrysalect violet, for which 0.25 grams of chrysalect violet dissolved in 100 ml of distilled water and 10% of glacial acetic acid 1 ml. Mix, filter and it is stable for 2 years in a amber color bottle. This is the procedure for Luxal fast blue stain. Deparaffinize the slides, hydrate to distilled water. Place the slides in Luxal fast blue solution at 60 degrees centigrade for 1.5 to 2 hours in the oven. Take out the slides and keep in running water for 1 minute. Differentiate with lithium carbonate and 85% alcohol solution equal amount for 5 seconds. Wash with distilled water. Repeat step 4 and 5 until there is a sharp contrast between the blue of the white matter and the colorless grey matter is distinguished. Counterstain with usine for 1 minute. Rinse in tap water or chrysalect violet for 1 minute. Rinse with distilled water. Dehydrate, clear and mount with DPX. Results: white matter blue, grey matter pink. Few tips for Llb staining. Best results are obtained if your sections are cut at 8 to 10 microns. Second one, if you preheat the Llb solution at 60 degrees centigrade in an oven for half an hour, the incubation time can be reduced by half an hour because the solution in coupling jar takes little longer time to get heated. That's why the preheating is needful. The third point is, while differentiating with lithium carbonate and alcohol mixture, a quick rinse in tap water will stop the differentiation. This is a good trick to prevent over differentiation. Phosphotungstic acid or Pth stain is used to demonstrate glial fibers in brain tissue and muscle striations in a muscle biopsy. First, we require Lugol's iodine, then 5% sodium thiosulfate, third is 1% potassium permanganate, fourth is 2% oxalic acid, fifth is PTH solution. Now, Lugol's iodine, 1 gram of iodine dissolved in 50 ml of distilled water, 2 grams of potassium iodide dissolved in 50 ml of water, and it both is mixed together and makes the Lugol's iodine. Second is 5% sodium thiosulfate. Sodium thiosulfate 5 grams dissolved in 100 ml distilled water. Third is 1% potassium permanganate. Potassium permanganate 1 gram dissolved in 100 ml of distilled water. Mix fresh, discard after use. Fourth solution, 2% oxalic acid, 2 grams of oxalic acid dissolved in 100 ml distilled water. Mix well, label with initial and date. Solution is stable for 1 year. PTH solution, 0.5 grams of hematoxylin powder dissolved in 50 ml of distilled water. 10 grams of phosphotungstic acid dissolved in 50 ml of distilled water. Dissolve the ingredients in separate portion of distilled water, the hematoxylin with aid of little heat. When cool, combine. Instant ripening of solution can be achieved by adding 0.1 gram of potassium permanganate. Make up the volume with distilled water up to 500 ml. Store at room temperature in an amber colored bottle. Let's look at the procedure for Pth stain. Deparaffinize the sections and hydrate to distilled water. Wash with tap water. Lugol's iodine for 5 minutes. Wash in tap water. 5% thiosulfate for 1 to 2 minutes. For glial fibers, bleach out iodine in 95% alcohol in place of sodium thiosulfate. Wash in tap water for 10 minutes. Oxidize in 1% potassium permanganate for 5 minutes. Wash in tap water. Bleach in 2% oxalic acid until white. Wash in tap water. Rinse with distilled water. Pth solution overnight at room temperature. Differentiate with alcohol. Rinse with distilled water. Dehydrate rapidly through 2 changes of each 95% alcohol and 100% alcohol and 2 changes of xylene and mount in DPX. Once it is mounted, check under the microscope. The glial fibers purple and the nuclei blue. Few tips and tricks for Pth staining. Best results are obtained when the sections are cut at 6 to 8 micron thickness. Second, to avoid precipitation, always filter the solution before use. Third, the differentiation step is very critical. You will notice the section is brown when you remove it from the PTH solution. Use 85% alcohol and keep differentiating till it turns purplish blue and stop. The Nusselt stain demonstrates Nusselt substance in neurons in brain tissue. Reagents required for Nusselt staining: 85% ethanol, crysyl ect violet 0.2%, acetate buffer and glacial acetic acid. The procedure for Nusselt staining: first, deparaffinize your slides and hydrate to water level. Next, you can flood the slides with the solution that you have prepared, which has 0.2% crysyl ect violet in acetate buffer with 2 drops of glacial acetic acid. Leave it for 15 minutes. Rinse with distilled water. Then differentiate with 85% alcohol. Dehydrate, clear and mount. After mounting, check under the microscope the Nusselt substance in neurons dark blue to purple. Few tips and tricks for Nusselt staining. First, the best results require thick sections of 8 to 10 microns. Second, always filter solutions before use to prevent precipitation. Third, when differentiating using 85% alcohol, do it quickly to prevent over differentiation.
Thank you, Mary Madam. That was a wonderful demonstration. I felt as if I was standing with Mary Madam and observing live. It was of great utility, I'm sure. But I'm sure this is in the kind of an experienced hand which is demonstrating. And in practice, we'll definitely have a lot of issues. So to address those issues, we'll have a small interactive session now, panel discussion, to ask some of the practical issues, doubts, clarifications. People who are online can ask by typing in the chat box. We'll take up those questions after this panel discussion. Some of the important ones only we are kind of want to discuss with you, ma'am. The first one is so many chemicals, so many reagents. So many chemicals, so many reagents are things have to be selected, then have to be used, have to be converted from a stock solution to working solution. Now, how to go about this? In the sense, do you recommend in-house preparation or some kind of ready-to-use kits or anything? So many chemicals are there. We always feel that something which is ready-made is better. What is your opinion on this?
Good evening, everybody. The three things matter in obtaining the chemicals. It is the reagents, the quality of the reagents, and the preparation, and the stability. So in-house, even though we have to prepare ourselves, but the shelf life is longer and it is less expensive. Example, we bought a Llb Stain. It worked for a week or 10 days. Later, we have to switch back to our manual, our own preparation. So the shelf life is very short when we procure, and it is very expensive also.
Thank you, madam. It's a good tip, actually. Now, another important question what comes to our mind is that in general, if you see the factors which affect the quality of any kind of work in the laboratory, like any kind of testing, there are pre-analytical factors, the analysis, post-analytical factors. So I would say there are a lot of pre-analytical factors which will affect. So what do you think is are some of the important pre-analytical factors which we need to take care that can affect the quality of the staining and the result outcome?
The first is the fixation, like the nerve biopsies, if it is fixed properly As soon as they take out the tissue, if it is fixed well in a buffered glutaldehyde or a buffered formalin, the results are very good. When it is not fixed properly, they leave it out for a little time and then they transfer it to the fixation, then whatever processing is done also, it's likely to flow. So preferably use a charge slide and fix it little longer time. If you are not getting a charge, earlier we used to use only egg albumin and we used to keep it for a long time. The overnight we used to fix it on the hot plate. The next day we were staining Pth Stain and it was coming out. Now, since we have a provision to procure charge slides, we can collect it on charge slides and we can stain.
Any storage instructions you recommend?
Storage, all hematoxylin should be stored in amber colored bottle so that the stability is good. And differentiation, that's another differentiation part is crucial, that when you differentiate, it's likely to float in KMNO4. So differentiate carefully and give 15 seconds to 30 seconds, check another microscope as I have shown it. Then you can proceed the procedure. It's not a problem that you can prolong the differentiation by doing it by 15 seconds, not at one time where you get over differentiated.
Yes. And control, how important is control?
Some of the stains we have an internal control, like Pth Stain, you have adipose tissue where it is, when it is get over differentiated, you know that it is brown in color, you are over differentiated. When it is dark brown, osmium color is taken up, then you know that you are differentiated correctly and you can go ahead with that. And control slides, when you collect it, then it's better that you collect it and store it in a box where it is dust free, because when you stain it, that can show as a precipitate or your staining default. So when you store it in a box where the dust free, then your stain is, you know that you are doing it on a right slide. And the second thing is always like lepra or amyloid, when you stain it, where the positivity will not be there in the whole site block. So you number the slides, number it as 1, 2, 3. When you collect some 50 slides, say if first time when you collect, pick up the first slide, the 20th slide, 40th slide, and the 50th slide. Run this control. Then you know that where it is strongly stained, and at the end of 50 slides also, if you can still collect, then you can store the block. When you know that the positivity is not shown that clear, then you have to discard the block and select the new one.
Yeah.
So when you collect 50, then first take the first 20, 40, and the next should be 10 slides, because you won't know where the positivity is. So you can store it like that and run the slide.
But do you think that every batch, every staining should carry control stain, both positive,
Yes,
negative?
Every stain should have a control, so that even you're sure that you're staining it properly and your differentiation is correct. When you don't run a control slide, then if your differentiation is too longer, you will not know that you have not done the proper job.
Okay.
Like whether it is under differentiated or over differentiated. So you have to run a control slide to confirm your stain yourself.
Correct. You usually check and then only release the.
I check the slide first, whether I check all the test slides. First, I'll check the control slide and to confirm my stain is correct, and then go ahead.
Correct. That's very nice tip. Now, in general, what we face in most of the staining is reduced intensity, suboptimal, or staining or not crisp or not clear. So, what are the main causes for understaining or weak staining, and what tips do you recommend to overcome that?
Some stains you'll have to collect thicker sections, say about 8 to 10 microns, like Nusselt, Llb for myelin, where the staining takes up darker. And the second is differentiation again. When you differentiate, you have to be very quick, because when it is alcoholic, then you have to stop with all water. If it is aqueous, you have to immediately stop with the alcohol base. So while differentiating. So when you use like Nusselt, when you use 85% alcohol, immediately you give a rinse with distilled water to stop the differentiation. Then you dehydrate with different grades of alcohol and clear and mount. Like if it is a aqueous stain, like Mason's trichrome, aniline blue is aqueous stain. So immediately you go to the running water, just rinse it to remove the aniline blue. Then quickly go to alcohol to stop the differentiation of aniline blue. So when it is an aqueous stain, you go to alcohol. When it is an alcoholic stain, you go to water to stop the differentiation.
Yeah, very important tip. Now, another problem what we face is when we do as kind of beginners like that, is either over differentiation or under differentiation. So we don't get optimal staining. So how do you monitor this differentiation, and how do you control and kind of regulate this differentiation? That differentiation, how do you monitor in the sense at the time of adding the differentiation, that particular reagent, whether it has been exposed to the proper time, or it's got excessive exposure and over differentiate has happened, or under differentiation? How do you monitor when you are staining this differentiation step?
That's what when you check under the microscope every time when you differentiate.
Yeah.
I always suggest that you under differentiate, check under the microscope, and then you go further.
Correct,
so
correct.
that your differentiation is optimal and the contrast will be very good.
I remember this. In food also, if you put under salt, you can always add the salt. If you add excess salt, then it's very difficult to manage. No, another important thing, especially for Pth Stain, is that most of the times when we do, it gets lifted. Because yours are experienced hands and gifted hands, I would say. So to overcome this lifting, not only in Kepal, in many situations.
The earlier fixation in nerve biopsies, it is only the fixation which matters and the processing. If your processing is not good, if the chloroform or the xylene, if it is over dehydrated also, it can the tissue will become brittle and it will not stick to the slide. So your fixation and your processing, if these two things are good, then your sections will be good. And Kepal, you collect little thinner sections, 4 to 5 microns. Don't collect thicker sections.
These are some of the questions which came to my mind. I'm sure Yashavani will be having some questions, and after that, audience, you can type in some questions. We'll ask that.
Yeah. Thank you, Mary, for that wonderful video. And I think now everyone can understand why you're such a good teacher. So step by step, you have explained it so well. And one of the important things which you just now highlighted, especially from a neuropath lab and coming to the nerve staining of Kepal, and that lifting is really a huge issue. And you've really highlighted the importance of correct fixation and correct processing. So thank you very much for that. And the other thing, just a point about the previous, some of the comments you made about checking the control yourself. I think you have highlighted with your experience how you confirm every time that your control has worked well before you submit the slides. I think this is one thing which you can impress that everybody needs to do. It's not just doing it and giving it, but checking that the controls have worked, which you always do and give us. Thank you for that. So you already mentioned about the importance of reagents in a good stain. So when we receive these reagents, this question I have is because whenever we receive, particularly from good brands and the same, cat number, we get the next batch and we just keep it and then we use it. But we do know that there are certain criteria for reagent acceptance. And is there any such thing as that you still need to, what is your advice and guidelines about these new chemicals or other new lots that we receive?
No, first, when you receive it, you'll have to check the seal, the company name and the seal, whether it is proper. Sometimes it can, you have to check for the seal. If the seal is intact, then you know that it is packed well and it is not open between or nothing has happened to the chemical. And the second one is you must look for the color of the, whether it is decolorized or discolored. Some chemicals, when it is exposed to air, when it is moisturized, it can change its color. So somewhere the leakage is there and you can always reject that chemical and procure a new one. And the stability also. And whenever you receive it, see to it that you run the slides with that batch and with the different controls where you have already done it, and check whether the stains have come out the same thing or it is different or whatever the changes are made. So you have to run the control with different, as I told you, that first and the 20th, 40th and 50th, and depending on that, you can run with 3 slides also with while doing it to confirm the stain.
So thank you, Mary, for highlighting the importance of checking every batch that comes and not taking it for granted that even though it's from a reputed company, there might be some issues with it. So every batch of reagents, so there should be some criteria for reagent acceptance physical, as well as the chemical. You check out the stain before putting it into use. So thank you for that. That is very important for quality control. And you are an expert in the stain quality control in our lab. So after doing all these things, are there any safety issues? What about us, about the technical staff safety? Any guidelines or any thoughts you can share with us?
Most of our chemicals, what we use is very hazardous. So we always have to use the mask, our lab coat, preferably if you're in contact with formalin or osmium, we have to wear a goggles. And always well ventilated place when you're preparing the solutions or when you're doing something, the lab work. And always, if possible, you can wear PPE also at times. If there is a provision that you have a fume, you can use fume while preparing osmium. And one important thing while preparing osmium is it comes as 1 gram. And you should not come in contact with your hands. Always, anyway, you'll be wearing the gloves, but still, you just break the vial, put it in a wide mouth bottle, add distilled water, leave it for a day, and don't shake it or dissolve it. Allow it to dissolve on its own, and the next day you can use the osmium.
Thank you for that, especially highlighting the dangers of the precautions that have to be taken especially for osmium. And now, after having gone through all these special stains and the importance of the technical staff and quality control, moving a little away, what about automation? Can this be automated and maintain the quality?
First, we must manually be thorough with all your stains and things before switching to automation. But one advantage with the automation is you get a uniform stain. Any number of batches you run, you will get a uniform stain. It's little expensive. Manually, the differentiation part is under your control. So you will have a control, whereas in automation, the differentiation will vary from person to person also. And some of our stains are very tricky, where you have to differentiate manually. And the volume, the capacity of the container, the capacity of your container in automation is not less than 250 to 300 volume. So you'll be using lot of stains. And some of the stains only you can use in automation, like mass and strike room, where you can reuse the stains. Where you cannot reuse the stains, we cannot use automation, because you'll be wasting 250 to 300 ml of stains, which is very expensive. Then
what about some issues like fixation issues? So that, when it's done manually, you can counter that by adjusting your steps. Whereas maybe that part in automation may be difficult. Like particularly you mentioned for KPAL.
Yeah, that is in automation, you can only, that incubation can be done in automation. The differentiation part, we have to do it manually. we cannot differentiate. Or else we have to give very short time, like 15 seconds, and check under the microscope and go back. But oxalic acid, again, it is okay, which can be reusable. And K-minophore cannot be used for a very long period, say about 3 to 4 days, because it's acidified. You cannot use it. Maybe 2-3 days time you must keep changing, which will be very expensive, 250 ml to 300 ml.
Yeah, so thank you, Mary, for highlighting both the positive points of automated staining as well as some of the negative points. I think for each stain that we select, we will have to weigh the
Wait.
pros and cons and then take it up. Right? That's a nicely discussed. Now, do we have any questions from the audience? I request Dr. Nandish to ask.
Okay, there is one question actually. You can clarify by typing if I'm not expressing it properly. They're asking, is there any special technique for frozen section H&D staining for neurosurgery cases? Special technique for frozen section H&D staining, like
Yeah.
generally we do a rapid staining.
Yes,
Do.
we do. And some fibrous tissue where squash cannot be made, we make cryosections, and we do the same rapid method. In 2 minutes time, we can finish H&D stain.
Some people have used Aldin blue.
Difficult to interpret. It is all blue in color, where cytoplasm also and the nucleus.
No, H&D special H&D staining.
Specifically mentioned.
Yes. Special technique for frozen H&D.
And then
can highlight that it's mainly
yeah. We don't do this cryosectioning for neuropathology cases, neurosurgery cases. It's more of squash, so that will save time for that rapid H&D. And one more thing happens is when you're cutting on cryosection, there will be some thickness like 4 microns, 5 microns. Usually that thickness you won't get on cryosection. You usually go up to 6, 8, 10 like that. But here, since it is squash, it will be single cell, maximum 2 cell thick. So the rapid H&D can also be made more rapid. So that will save time. Then I think if you're not like clarified with your question, you can type it more elaborately clearly. Now we can. The next question is: can PTAH be performed on muscle cryosections? So it cannot be performed. Is the opinion.
I will be answering this question. PTAH cannot be performed on the cryosections. We have tried it because you need to fix the tissue. Despite fixing the cryosections with formaldehyde, it did not work well. On the contrary, we have another stain which is called as the modified Gomery's trichrome, which will not only highlight the muscle fibers, but also highlights the connective tissue. So that's the only stain. We have also tried doing Masson's trichrome on the cryosections, but then it has not worked. So both PTAH as well as Masson's trichrome, please remember it does not work on the cryosections.
Next question is the strength of phosphorium tetroxide in Fleming solution. Is it 1% or 2%?
1%.
1%. Then, there are many messages from previous fellowship students, technician, technology students. So many congratulations are coming messages that only reflects the kind of contribution to the entire the fraternity. Really, this one feels good with these kind of messages and feedback. You're actually kind of overwhelmed. That also, one more thing I just want to mention is I think any number of books reading will not replace this kind of interaction with the experienced people, and very in a sense they are the best gifts, I would say, with whom we could work with and all those things. Mary Madam has worked for a long time and has been closely associated with Vani Madam, our senior professor, and Yasha Madam, our a professor of neuropathology. I request Vani Madam to express her views, thoughts, share her.
Good afternoon to everyone. I hope you can hear me well. I don't know how to begin to describe Mary. I'll start with her name. Okay, her name, Mary. Mary actually signifies devotion, it signifies sanctity, it signifies kindness. You know, in Hebrew, in the Jewish Hebrew, it says that it's a drop from the ocean. And I think that's what characterizes our dear Mary, who has worked with us for so many years in this department. I've known Mary. I think Anita has beautifully elaborated what kind of service she has given to the department. I've known Mary for more than 3 decades now. Ever since I joined the department, I think she was the first one to teach me techniques in the histopathology lab. As soon as I came, it was Mary and Kumar, all the time. And we learned so much from her expertise. And I've seen her grow in her, technical aspect. I'm not talking about special stains. Special stains is something that she's a master at. There is no doubt about that. But then every other technique in the laboratory, maintaining the laboratory, I think these are some of the highlights of Mary's work in our lab. She's worked with several faculty, several residents, several heads of departments, and she's worked with so many junior technicians. She's trained a lot of these, her junior technicians, and sure enough, she has set the bar very high, but I think she has passed on all her legacy to the juniors, and I'm sure they have learned, and they would be, I'm sure they're capable of taking this forward. And thank you for that, Mary. Now, if we talk about Mary's personality, I'm sure that all of you are very familiar with her wonderful laughter. Every time you talk to her, there's a little laughter that is there. And I hope Anita has recorded that laughter for us, for the department at least. So beautiful laughter, and that only tells us how cheerful she is all the time. But let me tell you that behind that, she's very thoughtful, she's very focused, she's precise, and she's practical. And that is what has made her give this exemplary service that we've had for all these years to the department, to take the department forward. She served this great institute, and in turn, it is all for patient care. We always receive the optimal stains, the optimal sections, and we are able to provide diagnosis. And finally, the end result is patient care. And like Nandish said, they're the pillars. I mean, they're without the technologists, we can't even, give diagnosis to the patients or help the clinicians. So these are some of her, her great plus points, I should say. And what I admire about Mary is her empathetic nature, the emotional support that she has given to people in the laboratory. It's just not that her technical colleagues or technicians, even to the hospital assistants, or even if it is a contract, person who's working in the lab. She's given fantastic emotional support to all of them. And we've seen that the way she supports each and every person. And I really love that, Mary. I really have appreciated that over the years. You're very fortunate. You have a lovely, wonderful family. And I think your upbringing is very nicely reflected in your children. Her daughter, Cynthia, came to work with me. Cynthia just walked out, but Cynthia came to work with me in the neuro-oncology lab. And I almost felt I had Mary next to me in the lab, so precise. And let me tell you, it was in the early days when Cynthia came, and she helped us to standardize the molecular technique with her 2 friends, little girls who came, and that was remarkable. I just loved having them in the lab. And I'm sure that, Mary, you're going to have a wonderful time with your beautiful family, your husband, your lovely children, your grandchildren, your trips abroad, coming back and looking after the kids over here. You have a lot of responsibilities, but you have to come back and advise the technical staff of the department, give all your wisdom and share it with them so that the department can be, taken forward. So thank you for all the service that you've given us, Mary, and we love that really. Thank you.
Now I request Yasha Madam to say a few words.
Hello, Mary. It's been such a pleasure to have been associated with you for the last 2.5 decades. From the time, your cheerful smile, your cheerful tone, your cheerful laughter just brighten up the lab whenever we walk in and we hear your voice. And of course, everybody has spoken about your expertise. It goes without saying. And it's not just the special stains, anything in the lab, we can walk in and we can ask you, Kumar, and it's not just only the stains. Anything else, you will have an answer, you'll have a solution, and you'll do the troubleshooting. And everybody spoke about your empathy, nature, all that is very good. But I also know about your firm nature. But you're very particular about the quality. And you're very particular that a thing is done, like Dr. Vani said, in a precise manner. So you brought that element of discipline and responsibility, which you coax and enforce the others also to follow. So you've led by example, and you're a high standard for everyone, all of us to live up to. And thank you for that. And apart from the technical expertise, I've also interacted with you, your empathetic nature. We have spent some time talking about sharing other things as well. And I think you've touched the lives of many people, of all cadres. And you have certainly touched mine too as well. And wish you all the best. I know you've got a lot of exciting things to look forward to with your family, or your wonderful, loving, beloved family. But don't forget your other family too, which you may have spent more time with.
Yeah.
So do remember us and keep in touch, and we'll keep bothering you with all our troubleshooting as well. And the other thing is, of course, all the well-wishers that are coming from everywhere, the numerous people that you have trained, just a huge thank you on behalf of all of them who are not able to say it, and from all of us in the department. Thank you and best wishes.
Thank you, Madam. I request Gayatri Madam to say a few words, because Madam has worked for a long time with them.
Hi, Mary. There. Last, so she's many people, I mean, Vani, Yasha, and Anita have told so much about her, and then I echo all those words that they have told. But I should say that she was also my teacher. She not only taught me special stains, but starting from cutting the sections, the paraffin sections, and then how you fix those paraffin sections, leave it on the hot plate. From there on, I began my journey in this department. Hematoxylin eosin staining, not just on paraffin sections, but also on cryo sections. You taught me Masons, trichome, you taught me even the PTAH, which I think even to this day, I cannot forget the use of that mordant, which is most important. So you didn't use this mordant, so you can't get this stain. So those are the words she used to tell. So she is very, very good at techniques. And after that, she took over even muscle enzyme histochemistry. So all of them have spoken about the special stain that she does for the CNS. But then she also has done a lot of enzyme histochemistry, and she's mastered even in those enzyme histochemical staining also. Now, that apart, she's a great friend of mine. So any trouble I have, any of the family issues that I have always discussed with Mary, and she is very humble and of course, very firm in whatever she stays. Then apart from all of these, she's also a very good cook. We all get the cookies I think all of you have forgotten about the Christmas cookies. So the rose cookies and the kalkals and the rose cookies. And she even taught me how to make it. And I'm so happy for that. Mary, I wish you all the best. and then enjoy your retirement life.
Thank you, Gayatri Madam, Yeshwa Madam, and Mani Madam for such nice, warm, wonderful words. Okay. I request Mr. Kaftan Singh Sehrawat from ICMLS, as President of ICMLS, as a representative of ICMLS, to speak a few words about this webinar and his thoughts about in general.
Jay, good afternoon, sir and ma'am. Special thanks and congratulations to Mary ma'am for wonderfully you have presented and explained everything. It's really a wonderful example for all of us, how we can work together in a team, how we can learn together, how we can work together for better patient care services. So special thanks to Anita Mahadevan ma'am and other faculty members of NIMHANS. Really, ma'am, this is a very encouraging initiative from your side. And we are now exploring all possibilities, how we can continuously work together, how we can continuously make such a initiative means platform for regular learning. This is a really a example for other institutions, how we can recognize the role of such dedicated technocrats, technical personals who are actually working behind the scene. And this is really again a very team efforts, team example. One sided there is a technical competency of our faculty members. Other side, one side there is a competency, clinical competency of our members. Another side we have technocrats who are working behind the scene. So both we can work in a team, we can learn in a team, we can contribute for these services. Thank you, ma'am, for giving this opportunity, and really we are very excited to work together in future as well. Thank you. Thank you very much.
Thank you, sir, for those nice words. We'll now have the felicitation of Mary Madam. I request Mani Madam, Yeshwa Madam, Anita Madam, everyone to conduct this felicitation.
Thank you, everybody. I had a good, memorable day today. I don't feel that it is my last working day. I still get connected with you all. Whatever I have experienced, I have shared with you. And nice to have you all with me. And the words which I was listening from you, maybe I was not realizing what I was. So when I hear it from you all, it sounds good. I'm very happy. Come. Come. Come, Hannah. Hannah. Hannah, come. wants to talk. You want to speak?
Actually, yes, from small, I've seen mommy always with her slides and microscope, and that always encouraged me to take that field. So being here, it's always she's been our inspiration and our model. So I would like to say thank you for being my guide for where I am now. And we are blessed that all her sacrifices these years is it's who we are now. So I would like to thank God for who she is for us. And thank you, mommy, for all the sacrifices basically that she did. So, yeah. Okay, I love you, Mama. Love you, Mama. Yeah. Say loudly, I love you.
I love you.
Mama.
Mama.
Okay, so with that, we have come to the end of this webinar. On behalf of the Department of Neuropathology, I would like to express my appreciation to Mary ma'am for her dedicated service to the department and also sharing with us her technical expertise in the field of neuropathology. I'm sure a lot of young technicians and postgraduates have benefited immensely from this webinar today. This webinar wouldn't have been possible without the guiding force provided by seniors in the department, Vani ma'am, Yasha ma'am, and Anita ma'am. So deep thanks to all of you. I would also like to thank ICMLS President, Mr. Kaptan Singh Serawat, for extending a warm support for this webinar. I thank Dr. Nandish for his informative talk, which was quite interesting. And we have all learned a lot about special stains today. I would like to particularly thank the staff and students of the Department of Neuropathology for their valuable contribution in conducting this webinar. Mr. Manu, who is not here, deserves a special thanks for his commendable job with videography. And it was just for 20 minutes, but it is the result of many hours of meticulous editing. Mr. Chandru from IT team has been the main person coordinating the smooth streaming of this webinar, which has enabled participants to attend this webinar online. Thanks, Chandru. I would also like to thank Mr. Manjunath, who has come here for photography. And we are also happy to have Mary ma'am's family with us, the cute little girl also here. So we are thankful to you. And last but not the least, my deepest gratitude to all the participants who have helped the webinar make it a successful event, both on-site as well as online. So with this, I would like to conclude this wonderful event, which was hosted in honor of Mrs. Mary Kas.
Thank you, ma'am. Thank you very much.
Thank you. We'll close the meeting. We'll end the session. Good evening, all.